today we're going to finish up with the small RNA phage. There's some really fascinating applications of some of the proteins that you find in these RNA phage. We'll talk a little bit about that, and then we'll do a review of all of that aspect of things. But you're all ready to take a clicker question, because I forgot to have them last time, right? OK, so let's go ahead. As usual, you have a minute to make your answer, and then a minute or so to chat with your neighbors about it, and then a minute or so to really decide what you'd like to answer. And to let everyone who's late show up. I know there are more than 14, 16 people here. So, back up, back up, back up. We have the technology. OK, we don't seem to have a consensus yet, so let's stop and have everybody tell your neighbor what you chose and why.
It doesn't make sense because it can't be code protein needs to be making a lot, so it's first. It doesn't make sense. Not wait a minute. Oh gosh, I think we got it wrong.
Panic? Panic mode? Rumor has it I'm supposed to restart when the level of conversation goes down, but it seems to be pretty level. But maybe that's your friends up here in the front row who are helping out with that. OK, we ready to vote again? OK, we're happy? Unhappy? Relatively speaking? Go.
I didn't read the question, so. The argument I was giving was.
Always a good idea to read the question, yeah.
Well, it's one of those.
Highly recommend it.
Answering the question.
Reading the question is usually a good idea.
Oh boy, you were just digging us into a hole right now.
I'm sorry.
Voted and they're sticking with it.
Are you going to reopen it, or
It's open right now.
It is open now. To you can trick it again each time. 4. Last chance. Push your buttons many times. Good. No more.
No. You feel alone.
OK, what do people think? We have some differences of opinion.
Oh boy.
We're not 100%, although most people are A or A and D. I certainly agree with A. Why?
Because you need it right away.
Code protein is what gets translated immediately. And so that you don't have any alternative secondary structures. And then everyone, or a few people seem to think replicase protein. How do you get replicase protein? After you have translation of the coat protein. So, and how is that regulated? Due to the formation of different secondary structures. So it's not the replicase protein. Maturation protein, nobody liked that. I don't like it either, because that's the secondary structure that forms when? After you have replicated the genome. Okay, so that leaves us with basically D and the lysis protein. Is this a... For 1 point. So how is the lysis protein translated? It goes backwards. Okay, yeah. What goes backwards? The ribosome moves back and forth. Is that due to secondary structure? Has to do with just dissociation of the ribosome, coming on and off, or not, as the case may be. And it really doesn't have to do, and particularly not with formation of an alternative secondary structure. So what does that mean? E is the correct answer, yes. Okay. So, many more questions on this? So the question is whether you can express the lysis protein immediately. And the answer is you're not expressing it immediately because there's no ribosome binding site next to where that particular start codon is. The only way the ribosome can get there is by moving back from where it's terminated relative to the coat protein. So sorry if I didn't explain that properly. But it's, yeah, one of the main things here is it's completely missing a ribosome binding site. So otherwise you would never get any unless the ribosome was already there. Make more sense now? Good. Okay. So we can hide these and move on. One of the things that we didn't talk about last time having to do with these small RNA phage is how they get out of the cell. Of course, we talked about the lysis protein just a second ago. That has to do with how the assembled virion, these are naked particles. They don't have any membranes at all, so they've somehow got to get outside of the cell. That would either be some kind of secretion process, or, as in the case with most of the bacteriophage, they just blast open the cell. And so they literally make holes. That's what these lysins do, is they just literally make a hole in the membrane of the cell. And so the virions can get out that way. The other big question, and we'll talk again more and more about this as we move through all the other viruses in the rest of the class, is how do you package the genome? And in this case, again, it's your positive strand RNA. You don't want to be packaging willy-nilly any kinds of RNAs. You want to be packaging specifically the RNA, which is going to be what you need inside of your virion. So the capsid protein actually binds to that alternative secondary structure in terms of the replicase, where you have the ribosome binding site for the replicase. The capsid protein will bind to that and block any more replicase, which of course you don't need at that point because you've made enough of it, and make sure that that gets put together. And I put here that it can assemble in vitro. This is one of the really nice things about a lot of the small viruses in general, but particularly the small bacteriophage, is that if you just make enough of the coat protein, you can actually make a capsid or a capsid-like particle. And that's where that structure came from that I mentioned at the beginning, where you said the maturation protein was hiding on the backside? So in that case, there wasn't a maturation protein around. It's just the capsid protein, and that's what they made the actual structure from. Just to finish up with these small RNA phage, yes, they're great examples for thinking about RNA viruses, which we'll spend quite a lot of time talking about for the rest of the term, but also taking advantage of some of the pretty unique aspects of some of these viruses. And this is particularly true for that replicase protein, the RNA-dependent RNA polymerase. The Q-beta replicase, and actually it turns out pretty much all of these small RNA phage, their replicases, the RNA-dependent RNA polymerase, is really, really good at making very, very large quantities of RNA. But it's completely dependent on all of those secondary structures which you have in the RNA genome. So if you can somehow figure out a way to get those secondary structures connected to an RNA that you're interested in, it's a way that you can make very, very large quantities of a very small amount of RNA. So it's sort of an RNA detection technique. And it's outlined basically here on this slide. So, oops, back up. I'll use my pointer now. There's this small RNA called the mini derivative 1 of your virus. It doesn't matter what that is. But it's a small RNA that can be made in very, very large quantities by this replicase. And again, it's got the required secondary structures, not surprisingly, at the 5' end and the 3' end of the genome that the replicase will recognize and then make many, many copies of. What you do in this case, again, this is a completely artificial construct, is you put a complement to your target RNA in this small RNA molecule, and that way, if that complement is around and you have this RNA, you can make very, very large quantities of it. And so the key is to figure out how you can get that target RNA. So your target RNA, let's just say for giggles that it's part of the HIV genome. So you're interested in detecting very small amounts of HIV. It turns out to be a major problem in terms of trying to actually finally get rid of HIV, because once it's infected, it's really hard to get rid of. So very small amounts of target RNA, then you get rid of all of the other RNA, and then you bind this to your modified MDD1, and again, it's got the complement to hopefully this target RNA, and you wash a whole bunch more to get rid of this if it's not bound, and then you amplify this like mad. So this is really, again, an RNA detection technique, and it takes advantage of the fact that these RNA-dependent RNA polymerases will replicate a small piece of RNA, again, dependent on that secondary structure, and any piece that happens to be attached to it. And so just in a cartoon form, it's like this. So the target RNA that we're looking for is this target RNA here in the middle. Again, let's say this is your HIV genome. First thing that you do is you immobilize this target RNA through usually DNA-RNA base pairing interactions. So you have this DNA oligonucleotide that has some kind of way of separating it from everything else. In this case, it's a magnetic bead that you use to connect it with a magnet. So your oligonucleotide magnetically separated, so you can take your cell lysate, whatever it is, which has some of this RNA in it, or it doesn't have some of this RNA in it, and isolate only that RNA, or, again, if there's nothing there, you're not going to see anything. But this amount of RNA can be like one RNA molecule in a really massive sample, so it's really going to be very, very hard to detect it. And so the idea here is with this MDV1 RNA, with your modified complementary sequence in it, you put this in, and if this target RNA is here, and the MDV RNA is there, plus your Q beta polymerase, it will make massive quantities of this and say, hey, yes, this particular sample is positive. It had that RNA in there. If that sample doesn't have the RNA, you're not going to get any amplification because this MDV RNA will get washed off in that second wash step. So this sounds great. It's wonderful. There's been a whole section in the textbook talking about it. What do people do these days? Janie, you here? Jane Cow? Didn't see ya. Do you know what people do now? If you want to detect small amounts of RNA, any guesses? You guys done this in the lab? Any other thoughts? Yeah, so if you want to identify one specific RNA, what do people do these days instead of this technique? Is there a particular RNA present or not? RT-PCR. What the heck is RT-PCR? Reverse transcriptase PCR. So you put in a reverse transcriptase, which we'll talk about much later, because that's what you see in retroviruses, and that will make a DNA copy, and then you make many, many copies of that. I think this is a cooler technique, and it actually works better, but RT-PCR is something that many more people use and seems to have kind of supplanted this particular technique. But it's a really wonderful way of making very large quantities. And again, it's kind of a review thinking about these small RNA phage because it's dependent on having that secondary structure in your RNA and being able to use it that way. So that's all we have for our small RNA phage. More questions, comments, worries? Let's go over to the other side and talk about DNA. So all we've done is taken off that 2' OH. So now we're a deoxy ribonucleotide. So single-stranded DNA bacteriophage, and this is actually only one class of single-stranded DNA bacteriophage. They're the so-called filamentous ones that we're not going to talk about. But these are the icosahedral ones. And as usual, we'll talk a little bit about where they were from. And I give you one guess. Forgot my list.
Sewage.
Exactly. Turns out that the single-stranded DNA bacteriophage is was a really wonderful tool for understanding DNA. And again, a lot like the RNA bacteriophage, where you need large amounts of RNA. Now, if you need large amounts of DNA, a defined sequence, it's a really nice way of doing that. One of the really bizarre things about these viruses is their replication. Single-stranded DNA. How much single-stranded DNA is made by cells usually? None whatsoever. So clearly this is going to be very different than the cellular processes. And then it's really well known how these particular virions get put together. Mostly the work of Ben Fain. And then part of the reason that I'm talking a lot about these is because it turns out you find lots and lots of these kinds of viruses in the environment. And I don't know if you remember from last week, like past the weekend, when we looked at the diversity of single-stranded DNA viruses, that massive tree with ridiculous numbers of branches on it, and most of them didn't have any kinds of names on them. Many of them are quite closely related to these kinds of viruses. So in the environment, you actually see many, many of these kinds of viruses. And in fact, we've discovered some of them as well. So everyone likes me to have a big picture slide. Again, nice thing to review for something that's happening in, say, about a week. Again, single-stranded DNA phage. The entry process here is actually brand new and even newer than the last version of our textbook. So we'll spend a little while talking about that. It was kind of surprising. The replication process, again, this is because we've got single-stranded DNA viruses. These are going to be very interesting. But the problem with single-stranded DNA viruses, and now I can get back to my list, is what, Helen Lee? Helen here? Nope, okay. Oh, I will completely trash this name now. Chanlianka Sok? Yeah. So what's the problem with single-stranded DNA? When you have single-stranded DNA that comes inside the cell, what's the big problem with that? Yeah, you have to have a second strand, exactly, before anything else can happen. So you have to have that second strand form. That's also called the replicative form of these particular viruses, so that RF. Then, to make your single strand, we have to go through this rolling circle process, and we'll spend quite a while talking about rolling circle replication. It's a very interesting process. It seems to be very specific for different viral genomes. And then the assembly process, we haven't really talked about this before. Turns out that FIX-174, which is the classic microvirus, is a really good example of the necessity for scaffolding proteins. And so these are proteins that are involved in making the structure, but are not there when you're finally done with the structure. And for FIX-174, it's actually pretty unique because it's got internal scaffolding proteins and external scaffolding proteins. And we'll take a look at that. And through that assembly process, there's something called the procapsid, which is made, and then after that procapsid, you end up with your final form of the capsid. So where do these guys come from? Again, mostly isolated in sewage samples, but also just from screening processes, looking for virus that infect E. coli, Shigella, Salmonella, etc. The best known of these is FIX-174, and most of what I'm going to talk about for the rest of the lecture is all going to be about FIX-174. Again, horrible naming here, but Phi usually ends up standing for bacteriophage. Here's another, Phi MK2K, etc. These were the original ones, FIX-174, and again, all these guys had infected E. coli and others. And it's only been much more recently that people have found these other bacteriophages, which infect a whole interesting set of bacteria, which are mostly obligate intracellular bacteria, or obligate intracellular, probably parasites or mutualists. One could argue about that. But it's interesting that these are now, the parasites of the parasites, or the viruses that are associated with the cells that are inside other cells. So it's this whole sort of Russian doll kind of approach to looking at things. And these may actually turn out to be the much more common version rather than the Phi-X174 type that was found in E. coli. And again, as I mentioned before, if you start to do metagenomics, you find many, many more of these kinds of genomes. So I mentioned where this particular virus came from. One of the ones, again, that we're not going to talk about, these filamentous single-stranded viruses, is called M13. I only found out relatively recently that M13 stands for Munich, 13th virus from the sewage treatment plant. So these are, again, very common places that you find them. A couple of things about Phi-X174, which is really interesting. Very early on, it was found that it was a quite small genome, only 5,400-ish bases of single-stranded DNA. The very first genome sequence of an organism, which they kind of push in the chapter, and I also like to remind people the first complete genome sequence was of a virus, not of a cell. So first complete genome sequence. And then I also wanted to mention here, mostly because there's this building. Anyone recognize this one? Probably not. No. UC Santa Cruz, actually. Something called the Sinsheimer Labs. It's not named because this particular person gave lots of money to the university, which is usually why buildings get named, but because he was doing really cool research. So you actually can get buildings named after cool researchers. So I'm waiting on that, but we'll see. So this is the Sinsheimer Labs. He decided that this was a really nice system to work with because it had a small genome and because DNA, and particularly DNA replication, turned out to be a very important thing that people were working on at the time. And having a defined DNA that you could work with turned out to be really important. So the template for all of the discoveries of DNA polymerase done by Arthur Kornberg, actually just up the road at Stanford, were done on Phi-X174 because it was a really good source of single-stranded DNA. But also, you could get the replicative form, which is a really good form of getting double-stranded DNA as well. It was shown after some of the beginning of Sinsheimer's work that that viral DNA just in and of itself was infectious. So you could take that DNA, transform it into a cell, and that DNA would turn out to make viruses. This was the very first case that that was shown for. And then it was made artificially with DNA polymerase in a test tube in the same year that the genome sequence was done. And so this was making life. People talk about synthetic biology now. Well, ooh, brand new thing. This was really the first synthetic biology, was in the 70s. So we're way behind the ball as far as that's concerned. And then there was a huge press release, et cetera, when Craig Venter's company synthesized the whole genome artificially, i.e. just chemically, but the DNA polymerase had happened many, many years before. And so this was a purely synthetic life because, well, okay, we could argue about whether viruses are alive, but chemically synthesized this particular sequence, put it into a cell, lo and behold, you make virus. So, yeah, nice, but again, it had been shown years and years before. So let's look at a bit of an overview of the life cycle. Again, life cycle of these viruses. So Phi-X174 genome comes inside the cell, and this is a single-stranded DNA. So again, as was mentioned earlier, the first thing that has to happen is you need to make this second strand. And we'll talk about how that second strand is made a little bit later on. This double-stranded DNA can be replicated to make more double-stranded DNA. Again, this is the RF form. But probably most importantly is this double-stranded DNA gets transcribed into RNA, which gets translated into proteins. Those proteins, turns out, are absolutely critical for making this single-stranded, because again, single-stranded is virus-specific, form of the DNA genome, and of course getting packaged and being released from the cell. So the fact that you have to become double-stranded and that this DNA is infectious by itself tells you that you're not bringing in any extra proteins. So even though single-stranded DNA, even in and of itself, can't do anything, apparently cells can deal with single-stranded DNA and make it double-stranded DNA, and so everything else can go on from there. So this is the overall process. Let's look and see what these DNAs actually encode. And this was one of the big surprises. Again, 77, very first genome sequence that was completed. The really bizarre thing, they saw one particular really bizarre thing, and in fact some people tried to call this an extracellular genome. So Xander, what do you think? Why do you think they called it, pardon? An extracellular genome. Xander here? Yeah. Yeah, so why did they think it was an extraterrestrial genome or a message from outer space? Okay, it didn't match anything, but it's one of the very first genome sequences. So that's not so likely. And in fact, this is what showed up in the New York Times. Examine tiny viruses from message from outer space. And this is literally a reprint from the New York Times. So this is, and not fake news. So why might they have thought that? Got a few more hands here. Jacob Brower. Repeating code? Overlapping sequences. And it was, at this point, it was very soon after Sidney Brenner published a quite famous paper that said, you can't have overlapping sequences. It's not possible. And then, lo and behold, there were these overlapping sequences. So this whole idea of being a message from outer space sounds really bizarre. But if you think about if there was an alien intelligence trying to leave us a message, something that would persist in the environment for long periods of time, that would be replicated many times, it actually wouldn't have been such a bad idea after all. Now, we haven't found who sent the message, so that's another story here. So what's one of the things that becomes pretty obvious? So this is the genome here. Probably the most extreme of these sequences is right here. You're actually using all three open reading frames. And so these open reading frames, again, you start at nucleotide 1. That'll give you sets of triplet codons. Start at nucleotide 2, gives you sets of triplet codons. You start at nucleotide 3, that gives you triplet sets of nucleotide codons. So Zephyr, Zephyr Kelly? Why is this such a problem? Why are overlapping codons not used in general, using multiple open reading frames? Hard to get the machine to process them, not so much. What happens if you make a mutation, like you change a single nucleotide? What could happen? It could change multiple protein sequences. So single nucleotides change multiple protein sequences. That's one of the reasons that people think that the genetic code is so degenerate and that it's not being used as an overlapping code. So what does this mean about these reading frames here that are all overlapping with each other? Joseph Ramirez? Picking everybody who's not here. Travis Spencer? I know that you're here if you've been clicking. Job or Joby Ritchie? Yeah.
What does it mean about them?
Yeah, what does it mean about these protein sequences? If, when we make single nucleotide changes, as Zafar pointed out to us, you're going to change multiple different protein sequences. What does that mean about these protein sequences?
They haven't mutated, so they still work, maybe?
They haven't mutated, so they still work. What's another possibility? They might have repair mutations, like extra repair machinery. That doesn't seem to be the case. Okay, we'll keep going. Travis? No, hang on, we go wrong direction. Armand Carasso? Yeah. Yeah, so so why do you think, what does it mean about the protein sequences if you can make, they've got overlapping open reading frames, and you make single nucleotide changes, you can, what does that mean about these protein sequences? It doesn't mean they've got extra repair machinery. It doesn't mean that it's problematic in terms of making them. So the translation could change. That's definitely possible. I don't think it's likely in this particular case. Any other thoughts? Courtney, Stout? So timing in terms of when these things are being made, that doesn't seem to be the case. Noof? Noof? I know I've got lots of hands up here already. Eventually I'll get up to one of them. Amy Lorber? Yeah, what? Hi.
Does that have to do with what you have on the slide, the mutation in some of what the overlapping things code for? They're not essential, so it doesn't have an effect on survival?
Or they could be really tolerant to mutation? Yes. So that is the answer. So sometimes looking on the slides helps.
Couldn't see it. You were standing right.
Oh, that's it. Okay, that's why. These excuses for the exam are not going to work. Yeah.
So
Nicole.
on that fact, are the ones that are essential, or that would be mutation tolerant, so B is mutation tolerant, would that mean that B is a higher conserved sequence or a less conserved sequence?
So what that would mean, again, just to sort of paraphrase your question, does it mean about more conserved or less conserved sequence? To have conserved, you're going to have other viruses that have that particular kind of sequence in them. And so then you could look at that conservation. I don't know that there are terribly many of these. Again, this is what people start to work with, and they spend their whole career working on one virus, and it turns out to be not a great example for a lot of other viruses. But the main thing here is it turns out that you can make lots and lots of mutations in that gene, and actually you can get rid of that gene completely, and the virus still functions. Yeah, Margot.
My question is, if it's a small genome and they have a limited amount of protein that they're coding for, wouldn't it be, like, I was thinking that maybe with mutation in one codon, because it's only making a certain amount of protein that they need, that they can tolerate the switch and still make some kind of similar protein that these are functional. My question is, correctly, if they have a small genome and they're making mutations in it, and how can they be tolerant if they're so limited in the proteins that they need?
Okay, so maybe I'm missing your question here, but correct me if I'm wrong. So to some extent, it has to do with the degeneracy of the genetic code. So single mutations, as long as you have a similar amino acid, are going to give you something similar. But it may also be having to do with the selection that you have, presumably, to have a small genome. Right. And if you have a small genome, and they have this really small genome, there must be really strong selection to have that small genome. Otherwise, you might want to make a bigger genome and then be able to have much more flexibility in terms of what changes. Because if you change one nucleotide and one amino acid in one of these overlapping open reading frames, it'll change a different amino acid in one of the other ones.
So we talk so much about how they have limited amounts of protein, and that's, they use the same protein for their capsid structure or whatever. So wouldn't making a mutation having a different protein really affect them?
Yeah, and so again, the question is, would it making a change make a really big difference? And it turns out that it is, there's probably, again, a lot of selection to keep a small genome. And these overlapping open reading frames, as we'll talk about a little bit later on, they increase the efficiency of production of the virions, but they're not absolutely essential, some of these extra ones. And so this seems to be a way that you can add extra genes. And so it's a way that these virions can take advantage of the fact that the genetic code has these multiple overlapping open reading frames and use some of these changes to maybe help a little bit, but at the same time, you have this danger of some kind of changes being a problem as far as some of the required amino acids which you're going to have. And that turns out to be this A protein right here, which is actually essential for your virus. So if you change too many of those sequences, it does turn out to be a real problem. Yeah, Nicole.
So I'm noticing that and this might not be the same for all viruses, not make the structural, the F through J, because there's something wrong that it knows it can't replicate, and that's why it splits it in two.
So the comment here is, as was very astutely noticed, that these proteins here are the mostly non-structural proteins. There's some exceptions to that as well, but these are mostly non-structural proteins, and then the rest of these are structural proteins. It's exactly right, and I'll get to this point hopefully in just a second here. Apparently, structural proteins, you can't have overlapping reading frames for, because if you could have overlapping reading frames all over the place, then you would expect to have them there too. So clearly, these proteins, you can't mess around with so much. The other question is, maybe if you have changes here and mutations that happen in some of these essential proteins for making more of the genome, then you're never going to see the rest of the virion. And that's exactly true. Is you have these, if you have a mutation which messes up an essential function, then you'll never see the rest of the genome. On the other hand, you could potentially have some mutations that change things a little bit down here, pardon me, that would then lead to a slightly different particle, potentially. Okay, so that's kind of a review of what we just sort of mentioned here. Again, we've got, oh, this thing, should have gotten rid of it. The structural proteins, again, are down here. And we've got these, again, these overlapping ones, particularly B here, is very well known as being not essential for function. As I mentioned just briefly, they've made mutants now, gotten rid of this gene completely. And it turns out that the E protein is also not essential. But they do stimulate the amount of virus which is formed. We'll talk more about transcription a little bit later on here. So this we had. More questions about 5X174? Everybody totally clear on it? Yeah.
So the overlapping reading frames is just another example of genetic economy.
Yeah, so the overlapping reading frames is an example of genetic economy, but a dangerous example of genetic economy because any kinds of changes there are going to potentially change multiple different proteins.
I was just going to say that having overlapping reading frames, you mentioned in the book that it might be an example of certain proteins being able to co-evolve with the host, and that people have nucleotide substitutions and being able to change along with the host.
Yeah, so the idea there, and again, this chapter was written by Ben Fain, who's a good friend of mine. And so the idea really here is sort of where do new genes come from? And one possibility, particularly if you've got a constrained genome, and why would these genomes be constrained, by the way? Get my next person on the list. Where are we? Oh, Margot, you're up. Why are genomes constrained? Sorry, that was a hint. Yeah, fit inside of the variant structure. So you can't actually expand your genome very much and still fit on the inside. And this turns out to be a real problem. And we'll talk a little bit more about that when we talk about the bacteriophages later on. Yeah, so it is, the thought is that this is a way you can find new genes. And just this overlap, and clearly you have a lot of constraints if you have an overlap, and then maybe there's something with the host that's also important from that point of view. But if you think about how viruses and hosts are going to be co-evolving with each other, the host comes up with a resistance to the virus comes up with a resistance to that resistance, and so on and so forth, the whole arms race idea. And so the thought is if you do want to change your genome, then that could be one way of doing it, is adding this extra gene by having these overlapping genes. Okay, now we can finally get to our next clicker question. And again, we've got a minute for everybody to first make your first guess, and then we can go from there. Come on. And we're going to get 100%, I'm sure, right?
Yeah.
And if we get 100% the first time around, then we don't need to have a discussion. I need some water. The only real reason we do clicker questions is so can I have a drink and a break. few more people showed up. 47. Oh, and I will try and upload clicker questions at the very latest this week so you can make sure that your clicker is appropriately registered. Okay, we could have discussions, but I think that since everyone's more or less agreed, that would kind of be a little bit of a waste of time. We agree? Or do we want to have 5 more minutes to talk to our neighbors? We'll move on. Okay, so yes, it is that they had these overlapping reading frames, and that's why people were like, this can't be true. It must be something that's non-isn't. And by the way, I think I did post that article from the New York Times on D2L if you'd like to take a look at it. It's a pretty quick read. Okay, so there are lots and lots of tables of virus proteins in this textbook, and people always ask me which ones are the ones that are going to be important, i.e. which ones are going to be the ones that are going to be on exams. So I try and add little things like red lines underneath the ones that we're mostly going to talk about. So the A protein in particular, the A protein is the protein that's most important for virus replication, those non-structural proteins, like the early proteins that we talked about. And we'll talk about how that works a little bit later on when we talk about the virus genome replication. The B protein is an internal scaffolding protein. The D protein is an external scaffolding protein. I mentioned this before. This particular 5X174 is one of the few that has these kinds of proteins. Now, the fact that we already talked about B being something which is very tolerant to lots of mutations means that that scaffolding can probably be a little bit more flexible in terms of putting everything together. Turns out that the D protein itself as well is also something which is quite tolerant to mutation. And then the H protein, which is the main protein which is required for getting the genome inside of cells. And again, that's something which is new as far as even the publication of our textbook is concerned. So those are the main proteins that we're going to be talking about here. The structure of these virions is a T equals 1 icosahedron, but it's a little weird. Normally, you would say, OK, T equals 1 quasi-equivalent icosahedron. How many capsid proteins would you have? 60. But it turns out you've got 3 different proteins, each of which are present in 60 copies. The F protein, the G protein, and the J protein. Turns out that these all really sort of fit together as basically like one protein, and then each of those forms pentamers. So you end up with 12 pentamers. And we'll see when we look at assembly, what that actually looks like when we look at the assembly process. So it's 3 proteins together. Each of them are going to form pentamers, and then 12 of those pentamers are going to come together in a T equals 1 like structure. And actually, I'm just going to put in the print job to make some of those particular particles a little bit later on. The next big question was that other structural protein, and that's the H protein. In our text, it says there are 10 to 12 copies of them. We think they're important for the virus getting inside the cell, but how? How are they important for the virus getting inside the cell? And these guys, as we mentioned before, are infecting E. coli. What's so hard about E. coli in terms of getting inside the cell? Wantrin? Mispronounced? Yeah. Why is it hard to get inside E. coli? How many membranes does E. coli have? Yeah, it's double. It's a gram-negative, so it's got an outer membrane and an inner membrane. So it's actually really hard to get through. And we saw with T4, what does T4 do? This syringe thing that goes in. But this is small. Where's the syringe? Where's that? And where's the tail? How do these things actually get inside the cell? So this was actually a pretty big mystery until, I think, well, let's see, when's it in this paper? Well, 5 years ago, so I guess it wasn't, that much of a mystery for that long. So Ben Fain, as I mentioned, is the fellow who has really done a lot of work on studying 5X174 basically for the last few years. It was his group that showed what the receptor was for these cells, and it turns out it's the lipopolysaccharide, which is also what you find on the external surface of E. coli. And lipopolysaccharide, not a protein. So this is our first example of a non-protein receptor. And it seems to be the glucose, which means that these guys can bind to pretty darn near everything because lots of cells have LPS on the outside. And then the question is, how does the genome get in? It was known that the G and the H proteins were important for this. And the spikes, and I should mention, go back here, take a quick look at the structure. These are the spikes right here. This is the G protein, the part of that trimeric protein that comes together as a pentamer. Makes perfect sense that this would be the case. But I was chatting with Ben one day, and this was actually right before the paper came out talking about the H protein. And I said, it's kind of like letting everything all hang out. And he said, you have no idea how right you are. And literally, about a week later, this paper was published, which was the structure of the H protein of Phi-X174. So this is the structure of the H protein of Phi-X174 while it's infecting its genome. So mentioned no tail, right? So if we go back, sorry to be bouncing back and forth a lot here, but where's the tail? No tail here. Well, it turns out that under the appropriate conditions, i.e. bound to LPS, there's this amazing conformational change that happens with the H protein, which makes these really long alpha helical structures. So this is 170 angstroms, so 17 nanometers. This is about actually I think the whole diameter of the virion is about 35. So this is about half the diameter of the virion. So the normal virion outside the cell, nice, wonderful icosahedron. When you have interaction with the cell surface receptor, this massive conformational change where all of these H proteins come together and literally make a tube that goes through the membrane, and the viral genome can come out through this tube. This was also shown here by cryo-electron tomography. It's a little hard to see with these individual cells. These are actually mini cells of E. coli. There's the icosahedral capsid, and then this is the tail that comes off of that, the H form, and this is various different molecules where you have this tube that's formed through both of the membranes. So normally, again, icosahedral particle, massive conformational change, where H basically inverts and makes a hole through the membrane. Yeah, Nicole?
Is it like a helix-turn-helix type structure that can actually appear and then open up and it's only on the spike, or is it part of the entire structure?
So the question, and I'll paraphrase here, where the heck does this H protein come from in order to make this amazing structure when you have the interaction with the membrane? And the answer is, we don't know. So if you just look at, well, maybe just look at the particle here. If you just look at the particle right here and just purify this particle as it's being assembled normally, there's no tail. And how the heck do you package this extra protein in here in such a way that when you have it associated with a membrane, it makes a tail? The answer is we really don't know. It was just really completely surprising that this was found because, oh, it's a perfectly normal, happy icosahedral particle. We know exactly how it gets assembled. But the disassembly process is through this, again, massive conformational change. Presumably, the H protein in here, when it's not associated with the cells, is just hanging out inside that virion and just waiting until it interacts with the cell and then forming this 10-stranded. And it's actually not a helix-turn-helix. It's more of a alpha helical bundle. And no one had ever seen a structure that looked like this before. I forget if it's 10 or 12. Yeah, so I've forgotten whether this is 10 or, let's see, we can count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So I think it's actually 10 rather than 12. But again, the question is, you have 12 of these five-fold axes. Now, why do you have, why don't you use 12? Why is this 10? Who knows? Again, it's biology, what happened. Now, how it happens, we can try and figure out later. So fascinating process and how it actually gets inside the cell. The other thing which was really interesting is if you look at the inside of this tube, and I know for sure that this was Ben's idea, that kind of looks like the mouth of a lamprey. And he says this is to make sure that the DNA, when it comes out, can only come out and not go back in. And so this is their cartoon. Again, where the heck is the H protein here? Who knows? Interacts appropriately, massive conformational change. We have the H protein making this tube, and then through the tube, you end up putting in your single-stranded DNA. So that's the entry process. Now the next question is, how do you replicate this? And again, it's a single-stranded piece. So how does single-stranded become double-stranded? Well, we always say single-stranded genome. Well, it's kind of like the single-stranded RNA that we have with the RNA bacteriophage. They're secondary structures. And it turns out that for all of these single-stranded DNA viruses, and my graduate student George Kaysen will talk about this later when he talks about the virus that he works with, they all have these secondary structures present in the genome. And this particular one is a hairpin structure, just DNA strand, loop, DNA strand, self-complementary. That seems to be a signal for the priming of normal DNA replication by all of the cellular machinery. So the DNA primase, the DNA helicase, etc. So you have the DNA primase, which will bind to the sequence here, and it's single-stranded already, so you don't need a helicase. And that will replicate its way around the genome together with the normal DNA polymerases. Here it has DNA polymerase 1. It may be DNA polymerase 1. It may be DNA polymerase 3. It's not entirely clear which one of those is the case. And then you ended up with this double-stranded replicative form of the genome. Replicative form, yes, because it's going to replicate, but also this now provides your template for transcription. And so the transcription, talked about before, one of the issues that we always have with these viral genomes, you know a lot more of the structural proteins than you do of the non-structural proteins. The way that Phi-X 174 seems to regulate this is really just at the level of transcription and translation. There are three main promoters in the genome, promoter A, promoter B, and promoter D. These are just the ones that are going to be transcribing each of these genes. So this transcript makes the A gene and maybe some of the other ones. Probably the strongest promoter in the cell is for this B gene, which tells you need a whole bunch of it. And again, these were our non-structural proteins, remember? Why are you making a whole bunch of this? Because you need a bunch of scaffolding proteins. Same thing is true of D, that other scaffolding protein. And then F and G, to some extent J as well. Not as much of the H protein. That's the one which is going to be forming this hole in the membrane. So because of these overlapping transcripts, you're going to have the most of the D protein because the B transcript and the D transcripts definitely overlap with each other. These are going to get translated. The A transcript turns out to be pretty unstable, so you don't make very much of that. And as we say, that's the protein that you need for replicating your single-stranded genome, which we'll look at in just a sec on the next slide. E, as we said before, is not required. H, you don't need that much of. Again, 10 copies versus the 60 copies. These guys are also regulated at the level of translation, so transcription and translation. The E protein has a pretty crummy ribosome binding site, so you don't have much ribosome binding. And both E and H have rare codon usage. What the heck is a rare codon? Anyone want to tell me? Jerkets? Yes, no? Brandon Page? Yeah. What's rare codon mean? So rare codons are not so much alternative codons or alternative stop codons. They're codons that you have very few RNAs for in the host. And so this is E. coli. E. coli has codons that it uses a lot and codons that it doesn't use very much. And those that it doesn't use very much, there aren't as many tRNAs around, which means you have less protein being made because you have less translation. So that also seems to be something which has been selected for in the evolution of these viruses. You don't have too much of the H protein, and you don't have too much of the E protein. So what is this A protein doing? This A protein I mentioned before, it's the protein that you need for genome replication, but particularly important for replication of single strands. So the A proteins are really fascinating proteins, and they should have been labeled here properly. They're just supposed to be this gray ball. They label everything else on here, but they don't label the gray ball. So what the A protein does is it will bind to your double-stranded DNA and make a nick, so a single-stranded break, in one of those two strands. And that single-stranded break is actually really kind of interesting because instead of making a break and having a free 3' end and a free 5' end, it actually just leaves a free 3' end because the 5' end gets hooked up to a tyrosine on the A protein. Tyrosines have OHs, so basically that OH is pretending that it's a 3' OH. Now you have the A protein bound to the 5' end, you have a 3' OH, and we've got all the cellular machinery. The DNA polymerase, single-stranded binding protein, and this is a rep protein, which is required for replication of Phi X174. What happens if you have a template and a 3' OH and a DNA polymerase? It's going to replicate. So the cellular polymerase binds to this 3' end, replicates its way around the genome. So this is our rolling circle because it just keeps going and going and going. Once you have one genome, which you've shown right here, and this genome gets back to where the A protein cut it in the first place, that A protein reverses its activity, takes the 5' end that was bound to the tyrosine, and relinks it to the 3' OH, which is here, and makes a new 3' OH for continuation of this process. So every time you go around, the A protein will make a new single-stranded copy of your genome, go around, new single-stranded copy of the genome, etc. This single-stranded copy of the genome can be packaged, or it can be replicated and made into this double-stranded form, the replicated form. Yeah? Yeah, so it actually reverses that activity. So it makes the nick in the first place, and then once the cellular DNA polymerase gets around to the other side, it will reverse that and religate that piece. That OH will now go and bind to the 5' end of the next piece and just continue to go around. So every time you go around, that A protein is going to reverse and make a new end. Yeah? Yeah, so the question is, just to repeat it here so it's on tape, is that are they single-stranded binding proteins from the virus or are they single-stranded binding proteins from the host? Did you see a single-stranded binding protein when we went through those genes, the genome? No. So single-stranded binding protein to the host. And as all of you remember from that course last term, one of the most common proteins that you find in any cell is a single-stranded binding protein. So there's plenty of that around. It's going to bind to the viral genomes as well. Yeah? How long does this process take going around the genome? And so again, it's a 5,000 nucleotide. The actual replication process is incredibly fast, but the whole process from infection to producing new virus particles takes about 10 to 12 minutes. So it's not just this replication, but it's the injection, it's the double-stranded formation, it's the transcription, the translation, the formation of single-stranded DNA, and packaging, which again takes from 10 to 12 minutes. It's crazy how fast this happens. Yeah, Jacob. I'm not sure I quite understand your question, but he means in terms of the oligomerization process. So once you have, and again, correct me if I'm not understanding your question here, but the idea is it's whenever you have a single-stranded binding protein, it's going to be a cooperative process. So one binds and a whole bunch more are going to bind. Yeah, that varies from single-stranded binding protein to single-stranded binding protein. The E. coli ones are different than the eukaryotic ones in terms of the stoichiometry and how they bind. So, but the stoichiometry here seems to be just the same as you have in cellular processes. Okay, so this is going to lead us to our next clicker question that everyone's going to get right on the first round, right? Okay, so let's do this one. Go. And I even capitalized one of them. And that was a great question leading into this. Could have had SSB on here. But didn't add that one. That's F, which is not in your clickers. The other thing I need to remember is to plan more time for clicker questions and less time for slides and lectures. Five. Okay, so whoever thought DNA Pol 2 and the B protein can come up and talk to me after lecture. So the answer is yes, the A protein. The answer is A. How useful. Yeah, sure.
Both the A and E sometimes use like really crazy complex proteins. Are they quite big compared to the proteins you're working with?
So size-wise, A and H. Actually, H is pretty small. So if you look at the genome, the H protein is relatively short. And the A protein is really quite big, but not ridiculously large in terms of some of the other proteins that we'll talk about later. But yeah, not particularly large one way or another. Okay, so I want to finish really quickly talking about assembly. We've kind of talked about this already a couple times. We've got two kinds of scaffolding proteins. We've got an internal scaffolding protein and an external scaffolding protein. Again, there's this internal scaffolding protein that can have all kinds of mutations and still be functional. External scaffolding protein, the way that these come together, F protein and G protein. Remember, you have 5 each of these proteins that stick together as pentamers. Those require the B protein to make these pentameric structures. Once you have these pentameric structures, then you have the D protein on the outside. All of these come together to make the procapsid, and the procapsid is this form here that's just proteins. The next thing that has to happen is we need to put the DNA into this procapsid structure and eventually get the final form of the virion, which we'll talk about on Wednesday.